right, so now we will move into the discussion from Dr. Kleefstra. So Dr. Kleefstra, are you there?
Yes, I am. So
wonderful.
Trying to share the screen now. Let me see. Do you see the screen?
We do.
It's a full presentation, or?
Right now we have you and I spotlighted, but we'll spotlight you alongside the presentation.
Okay. Okay, so thank you very much, Jeff and team from iDefine and the Boston team for organizing this this webinar. And I'm sorry I was a little late because of again this time challenges and change, but we are there, and I'm glad you already heard the great stories from Joost and Neil. And I thought to add on that and give you a flavor of several other studies or projects that are ongoing in our team. And I think with the 10 years experience that we have now in following up cases and including novel cases that are seen in our hospital, we gradually get some experience to have a more systematic approach of several angles. And so we are based in Nijmegen, as you've already heard, in the Radboud UMC, that's a university hospital. And there, for Kleefstra syndrome and many other syndromes, we have a great clinical team, and this is embedded in what we call the Expert Center for Rare Congenital Neurodevelopmental Disorders. And basically, there is 4 large angles that we approach for Kleefstra syndrome and other syndromes, and that's from the medical side, so the clinical part and clinical genetics. It's important. The biology, so we try to closely work and connect to underlying biology, as Neil just showed very nicely by studying these cells from the patients. And we closely collaborate with psychologists and psychiatrists to understand the developmental issues and neuropsychiatric aspects that a lot is common in these syndromes, and of course also the Kleefstra syndrome. And by integrating the knowledge from these fields, we try to optimize the personalized strategies to get better management and intervention for your children and the patients we see. And we are happy to have this great team and wonderful, bright, young researchers, clinical researchers among our team, and I show you here, and I will pass them during the presentation. And I think Joost here on the left, you have already heard, and I will not further comment on his studies, but I will show some other studies, and it might be some difficulty there or technical difficulties, but I try to pass you through, and there might also be time afterwards to discuss. So focusing purely on the molecular aspects in genetics and related to the clinical part, I'll first show you a glimpse of the studies that Dimitris is conducting. So you may probably aware of, have heard about it, that the most of the variants we identify in ETMT1 in the gene by our diagnostic approaches, they are one type of variants and called loss of function. That's a typical, the vast majority of variants in ETMT1 causing Kleefstra syndrome belong to that category. And until Dimitri started these studies, there were only a few missense reported. So that's another category, and we call them missense changes, published in literature or known to us, and that were in fact only 3. But by the current day's technology, so meaning the whole exome sequencing or whole genome sequencing strategies, there's many such missense variants identified, and we receive a lot of questions from worldwide from colleagues or patients or parents that send us a message about a variant with uncertain significance, in short, VAS, and that mainly or mostly confined to these missense variants. So we started to collect this and approach that by a systematic manner and try to learn from the spectrum of VASes that is now out there and known to us. You can see schematically how the gene, E71 gene, is composed of exons. So the exons are all these vertical bars, and this comprises the entire E71 gene, and it's located in the tip of the chromosome 9 Q arm. So this is the entire chromosome 9, and this is the Q tip. This is E71, and this is in fact also you see here all the exons with the vertical bars, but here's also protein domains schematically presented, and on the top you see the different variants that were known to us in 2012, so 10 years ago. So we collected missenses, a lot of such VASes. You again see the schematic presentation of the protein of E71 with the protein domains colored here, and above you see the different missenses that we collected. So we collected molecular information, but on top we also collected clinical features, of course, and we used 3D protein modeling. So how, because E71 protein is not a vertical bar or a horizontal bar, but in real life it's really a 3D, makes a 3D structure, and variants, E71 variants or mutations can change such a structure. And we used an additional test. I'm not sure some of you have heard of it, but it's called the DNA methylation signature. So we can look at different signatures on top of the DNA sequence. We can also look at how DNA is packed with proteins with a specific category, and that's called the DNA methylation. And nowadays it's known that for E71, for Kleefstra syndrome, there's a specific DNA methylation signature, and we can ask the laboratory to look at it, and that can confirm the diagnosis. And this study we did in collaboration with Bekin Sadikowicz from Canada. So for this 29 missense, we used these 3 approaches, and in the end we could provide for 26 of them concordant results, meaning that we could classify 9 as benign variants, are not causative for the problems, no Kleefstra syndrome, and 17 of them were definitely pathogenic, causing the phenotype and underscored the Kleefstra syndrome presence in the cases. But 3 variants were discordant, so were not in line with Kleefstra syndrome and not with the DNA methylation signature, but neither were classified or could be classified as benign. So for these 3 variants, we needed additional confirmation, and for that we started collaboration with a Japanese group. I'm also happy they are here today. They will present not on this work, but they also carry out this real great functional assays, and they have taken that up in their essays and more look by a biochemically manner and see whether these variants could provide pathogenic effect through different means, actions, than the classical E71 missense or loss of functions. So that's an ongoing story, but to summarize this part, so the variant interpretation currently, variants that derive from whole exome or whole genome sequencing can be challenging, and it's not always easy to classify them, but our data, I think we are now quite comfortable that we can combine the clinical input with the 3D protein modeling studies and the DNA methylation studies, and probably functional assays for those that cannot be classified by the combination of the first 3 means. And we also have indications that there is different functions for E71 on top of the histone methylation that is usually thought to have a major function. So that's on the clinical, genetic, and molecular aspects. Then another study, and that's in fact coordinated by Ariane Bouwman, another bright PhD, started in our group last year, and she will also be a PhD working on this brain model that was just presented. And she's more looking at clinical profiles, so looking at the EEG measures, but also measures that can be done in the body composition, in blood, in urine, looking at metabolism, etc. And for those who were present in the webinar 2 years ago, our pediatrician Joyce Gelen presented the study on the growth, and this is just a summary of the findings by that time, where we have seen that the growth is typical for this syndrome. So this is a plot, the age on the x-axis and the y-axis is the standard deviation. So it's a statistical way of showing data. And here in gray, you see the mean from our Dutch population. And here, cases start to grow, but you see there is, if you look at the height, that's this curve, height increases, the standard deviation it makes a short top already pre-puberty, and then compared to the mean Dutch population, there was a decrease in height. If you compare, it's relative, but the weight behaves differently, in fact oppositely. So there it gradually increases with age compared to the mean in the Dutch population. So of course, for the individual cases, it can be different, but as a group, as an entire group, the Kleefstra syndrome population has a relatively shorter, relatively shorter in height and larger in weight compared to the mean, and thus has also increased BMI at puberty and adult age. And because of this growth patterns, we also started to more systematically measure other functions as thyroid functions, and also the X-ray to define and evaluate bone age that you can read from X-rays. And what we see quite frequently, I must say, is a defect in thyroid function. So this is a very schematic representation of how the thyroid functions in our body. So the thyroid produces this thyroid hormone, and it's under influence of the thyroid stimulation hormone from our brains. And when the thyroid hormone it decreases, the thyroid stimulation hormone increases, and the other way around to get this feedback loop correctly. So when we find a low thyroid hormone, then usually then you would expect a high TSH, but we see in several cases that it's not that clear. So we see more a normal TSH or something on the high normal range, but not that increased as you expect. So, but we do see quite several of the patients a low thyroid function, and we need to supply the patients with the thyroid medication. And regarding the bone age, so in those cases there we started to have bone age measured, and there is this automatically measurement that our radiology department has, and I think many university hospital will have. It's called Bone Expert. But we not only saw a bone age that might be some ahead of the biological age, but we also saw in quite some cases a low bone mineralization and also some mild bone abnormalities, and that warrants us now to provide and to make in every child such hand photography. We in the past we did not do that in a systematic way, but we do now, because it might be relevant, give relevance for the clinical guidance and clinical follow-up. So to summarize this more or less clinical part, so we call it the clinical characterization and do it in a better and more specified and systematic way. And Ariana Baumann, so the young doctor, PhD studying this, she is very happy if you would connect to her in case there's any abnormality found, or if you have questions on these measures or would like to have such measures done, and we can see if we can be of any help there. Then the biology is just shown, not very nicely presented by Neil, but it's great work he's doing on the cellular models. In Nijmegen, there's also this great work done by Annette Schenk. She's studying fruit flies as a model organism, also for Kleefstra syndrome, many syndromes. And one of the projects that we are doing together with Annette is on sleep. And that project is done by Lara van Rensen. And sleep is very difficult to study. There's many aspects that play a role in sleep. But it did also pass during Joost's talk, where sleep might be a precursor for severe neuropsychiatric problems. But of course, young children might also have sleep difficulties, and it's often very complicated. But one of the questions is whether the biology behind sleep plays a role. And that might be true for each MT1. So for the Kleefstra syndrome, yeah, the biology, so the processes in the brain might be underlying at least part of sleep problems. And we know that might be present in other syndromes as well. So that is, Lara is focusing on sleep, and she studies that heavily in the Drosophila, because fruit flies also sleep. You may not be aware, but they do sleep. They have a rhythm. And in fact, this is the setup that can be studied in using fruit flies. So here you see the 24 circadian reflection of the circadian rhythm, 24 hours, and the bar diagrams that are indicated, measured by sleep per 30 minutes. And when gray is normal, you can see that in the yellow line reflects severe problems in sleep and showing less sleep for that specific fruit fly model that has been studied. And there is many parameters that is comparable that can be studied in Drosophila and that we study in human as well. So comparing sleep parameters and sleep trajectories for fruit flies might give us information on the biology for our human situation, which is obviously much more complex than such a small model organism. So Lara is conducting sleep studies in different syndromes, and among them the Kleefstra syndrome. And we ask the Dutch parents who participate to have this calendar filled for 2 weeks, and also a sleep questionnaire to be filled in. And for those that appear clinically appear abnormal on the inventorization of these data, that we can follow up if needed in the sleep clinic that we collaborate with in this study. And of course, it's not always sure whether your child is asleep or awake. If you sleep yourself and the child doesn't make that noise, you're not completely sure. And therefore, we extend the studies now with measures like the motion wards and an MFIT, which can be put under the mattress during sleep, and can also measure all kinds of parameters. So also here, we for some sleep measures, we could easily include people remotely by sending out questionnaires and sleep calendar, and we have given consent for that by our ethical committee. So if you would like to learn more about it or to have participation, you can send an email to this message. Well, then Neil already nicely presented the work. So that's the ongoing study that we just started, this great effort to really trying to combine this multi-level approach for the individual patients measuring the EG, but on top of that, we need a detailed medical history, physical examinations, neuropsychiatric assessments, and the measures on the metabolism, because all these measures influence, of course, the condition of the individual. So that we try this in this huge effort to combine. Then all studies and all the experience and all the data that we collect and get knowledge about, I think we are there now to head to get it more formalized in a guideline. And we are happy that we are supported by the European Union to develop such a guideline. I think there's around 55 professionals who are really active participating. And I'm also thankful to IDFine, who organizes work group meetings for such a guideline. And there the aim is to get this international standardization and recommendations for diagnostics, the care and the management for Kleefstra syndrome. And also here, I really need to thank Ariana Baumann, who's really taking the lead, the clinical lead to get the work done. And I also put the rest of the juniors over here, or who all have a very important role in getting the data and getting the work done for this guideline. It's really a huge effort that we need to do together. And I also like to thank Joyce Galen, the pediatrician, who is tightly involved in the care of these children in our hospital. And with that, I would like to give the floor back to you, Jeff. Let me see if it works. Yes, there I am. Not sure if Jeff.
I'm here. I'm having a little bit of technical difficulty. And there we go. So thank you for that. And now we can jump into the chat for a couple of questions real quick. And they're pretty wide ranging right now. So let me start with one question here, Dr. Kleefstra. It said, can medications, particularly olanzapine, be responsible for the weight gain in some of the older KS patients? Dr. Kleefstra, I think you're on mute. Sorry.
Yes, you're right. Yeah, so in general, it is known that olanzapine or antipsychotic drugs make you vulnerable to increased weight. But in our experience, because we also give clear instructions on the food intake, we not see so much weight gain on olanzapine. So, and also the curves I showed were corrected for olanzapine or antipsychotic weight increase. It's just intrinsic. It's really intrinsic in the metabolism of the syndrome.
Great. Yeah, I think diet is a whole nother kind of can of worms that we could get into. Diet and GI and
diet.
And again,
I had only 20 minutes. I think I already passed it. I think so. We also studying gastrointestinal problems, the diet, indeed, how it's called, microbiome, and there's so much to study. And cardiology is also very important. So, yeah. So this I would just give you a flavor of all the work that's done. It's really a lot of work done, and I think that's the most important message.
Yeah, I think just to that point, there is so much going on throughout the kind of Kleefstra syndrome effort right now. It's going to be hard to really tap into all of that today, and hopefully, these conversations, like the one that we held last year and the one that we held this year, is kind of giving you a high level overview. But there is a cadence of communication that we want to develop via our social media channels and via our newsletters, where we really ping the community about getting a little bit more granular in more webinars, and whatever those touch points can be. We are open to providing that communication and want to provide that communication. So whatever the kind of relevant topics are for the community, we want to be able to touch base on. So one other question real quick. I'm going to try to combine a couple of these questions. One is regarding is there a correlation between bones and teeth in terms of thyroid function? I've actually seen quite a few posts recently about teeth and the enamel being soft, but also whether or not calcium supplements could potentially be utilized to help offset osteoporosis.
Yes, they might be favorable. It's difficult to have a general recommendation at this point, or to dive into individual cases. But I think my message, just be aware. We recognize there might be bone problems, and not necessarily clinically already obviously. So there need not to be fractures, for example, because you might expect there might be bone fractures or increased bone fractures, but I don't think that's our impression. But yeah, there might be real to severe mineralization problems, and that's just check with your doctor and make an extra X-ray if not yet done.
Yeah, and then so I think we can wrap up this discussion for now. I think maybe it's more of not a wrap up, more of a pause. And then again, a lot of these questions, I think what we continue to find is that we are a rare condition where there's still so much to be known. And, that's going to segue later into some of the data collection efforts. I think that from our community, or from our perspective within the organization, we talk about data a lot, and it's because we want to really collect all of this information to be able to start really formulating a stronger idea of what's taking place, and not having these kind of one-offs, but understand what are the overlaps, what are the themes, what are the things that we're consistently seeing. And without patient population contributing to that through these data collection efforts, then we're never going to get to the point where we need to get to, right? So it's really an intertwined kind of relationship where it's so much, it's very reciprocal. So thank you, Dr. Kleefstra. Please just keep your eye on the chat for me, because I'm sure there's going to be quite a few questions coming in for you.